Alright, so let's get into glycolysis. So, glycolysis is the process in which the glucose molecule is broken down to form two molecules of pyruvic acid. As we can see here, the pyruvate is broken down to form the carbon molecules. So, the two sets of pyruvates left combine with the coenzyme to form acetyl CoA. So the first phase of glycolysis requires energy, while the second completes the conversion to pyruvate and produce ATP and NADH for the cell to use for energy, as we can see here. So overall, the process of glycolysis produces a net gain of two pyruvic molecules, two ATP molecules, and two NADH molecules for the cell to use for energy. Following the conversion of glucose to pyruvate, the glycolysis pathway is linked to the Krebs cycle, where further ATP will be produced for the cell's energy needs.
So glycolysis can go through an anaerobic or aerobic process, but let's say for example that it goes through an anaerobic process. So with that said, it would need more NAD+. So from glycolysis, we have 4 ATPs with a net of 2, and then 2 NADH. So with fermentation releases some of its NAD+ to NADH, and it drops off some of its electrons, which then creates NAD+. And then with that, it would then create two alcohols, that depending on if it's in a plant or an animal. So for a plant, it would go through alcohol fermentation, where it would produce 2 alcohols and 2 carbon dioxide molecules. But if it's in an animal, it would create 2 lactates, and it's through a process called lactic acid fermentation. The Krebs cycle, also known as the citric acid cycle, occurs in the mitochondria. We see here that glycolysis processes in the cytoplasm of the cell, and following is the intermediate step of the Krebs cycle in the inner membrane of the mitochondria, and this cycle finishes off in the matrix of the mitochondria. So this intermediate or half step practically prepares all of the necessary molecules for the main step of the cycle. So in the membrane of the mitochondria, the two pyruvates from glycolysis needs to have oxygen present in order to oxidize through the aerobic process. When the 3-carbon structure breaks off and oxidizes to create carbon dioxide, and this small process occurs for each of the two pyruvates, which gives us 2 carbon dioxide molecules. Coenzyme A attaches itself to the 2-carbon molecule, aka acetate, which then processes acetyl-CoA, which is one of the fundamentals for the next step of the Krebs cycle. Within this intermediate step, we also have NAD+ molecule from each pyruvate that binds with an electron and hydrogen proton ion that allows it to become NADH. So with this said, we are left with 2 carbon dioxides, 2 acetyl-CoA, and 2 NAD+. NADH. Following through into the matrix of the mitochondria, acetyl-CoA binds with a regenerative molecule called oxaloacetate, which makes this molecule regenerative is because there is such an abundance of the molecule in the matrix. There's no limit to how much it could be used, unlike the acetyl-CoA. So the acetyl-CoA binds with the oxaloacetate and completes this magical process. So the end goal of this whole entire process is to get rid of the carbon and create and store as much electrons as it can for the next step in the electron transport chain. So the first step was to rid of the 2 carbons by oxidization, creating 2 carbon molecules. Then through substrate level phosphorylation, 1 ATP is created, 3 NADH, and 1 FADH2. This cycles for 1 acetyl-CoA, and because we have 2, we will cycle twice. This results in a total of 4 carbon dioxide molecules, 2 FADH2, 6 NADH, and 2 ATPs. And because 2 ATPs is obviously not enough for the ideal optimal 38, through cellular respiration, we go on to the electron transport chain, where we're able to make the other 34.
Okay, so let's get into the electron transport chain. So this is a cluster of proteins that transfer electrons through a membrane within the mitochondria, as we can see here, to form a gradient of proteins that drive the creation of adenosine triphosphate, or ATP. So the electron transport chain releases energy stored within the reduced hydrogen carriers in order to synthesize ATP. This is known as phosphorylation, as the energy to synthesize ATP is derived from the oxidation of hydrogen carriers. So this occurs over a number of distinct steps. However, first you have to generate a proton motive force. The hydrogen carriers, NADH and FADH2, are oxidized and release high energy electrons and protons. Then there's an ATP synthesis versus I almost messed this word up, chemiosmosis. But the proton motive force will cause H plus ions to move down their electrochemical gradient and diffuse back into the matrix. So as the H plus ions move through the ATP synthase, they trigger the molecular rotation of the enzyme, synthesizing ATP. So then we move to step 3. This is where we have reduction of oxygen. In order for the electron transport chain to continue functioning, the de-energized electrons must be removed. Oxygen, in a way, acts as the final electron acceptor, removing the de-energized electrons to prevent the chain from becoming blocked. Oxygen also helps with binding the free protons in the matrix to form water. Removing matrix protons maintains the hydrogen gradient. In the absence of oxygen, hydrogen carriers cannot transfer energized electrons to the chain and ATP production is halted.